Hello, everybody, and welcome to this episode of CMN Live. My name is Karen O'Hanlon Cohrt, and I'm delighted to be joined today by a special guest, Xavier Duportet from Eligo Bioscience. Before we get started, I want to apologize for the delay. Thank you all for your patience, and hopefully it will be worth the wait. So, Javier, welcome to this episode of CMN Live. Delighted to have you here today.
Great to be here.
Yeah. So today we're going to talk about the microbiome, a little bit about what the microbiome is, how it's involved in health and disease, and most importantly for our conversation, how gene editing can be used to open up, I guess, new ways of treating human disease by targeting the microbiome. So why don't we start first with the basics? You hear a lot about the microbiome. It's a bit of a buzzword. It pops up everywhere in TV ads, in the popular science media. In your own words, what would you say the human microbiome is?
So, at least for us, in the way we're looking at it, is that it's the trillions of bacteria that are inhabiting our body, whether it's on the skin, the lungs, the guts, the vagina, and also, our tumors. I mean, our tumors when we have them, unfortunately, or even organs like now we're seeing we have bacteria in the brain. Or so basically, it's all the bacteria that are living with us, most often which are beneficial, but in some cases actually which either trigger or drive disease progression.
Yeah, great. So just on that note about the human microbiome being such a big buzzword, in the last 15 years or so, huge efforts have gone into understanding in some detail, I would say, exactly how the microbiome impacts not only our health, but also, how it is playing into certain diseases. One of the biggest initiatives that I am aware of is the Human Microbiome Project that started maybe almost 20 years ago now, or maybe even before that, really conceptually, with huge amounts of money gone into that. And there have been some findings from that already. What would you say have been the most important outcomes of those studies so far?
Well, I think there are a couple of outcomes. I think at first, well, this project, the Human Microbiome Project, at least helped. So there was a lot of basically sequencing done at different depths to try to understand how variable. So what was this microbiome? What are the species that are composing the microbiome? How it differs based on body sites, and try to understand if there is like a core microbiome, which is quite ubiquitous. So if we can find the same microbiome composition across individuals, whether it's across the globe, whether and different populations, different ages. And I think one of the key finding was that yes, there is a core microbiome. So there are some bacterial species that are very often, most of the time, found across individuals. But more importantly, I think beyond just the fact that there are these core species being presented, it was that you can have some strong variability in the microbiome composition in terms of bacteria, but in terms of genes and functions, you can have a very, between two persons, you can have a different microbiome, but the functions that are carried out by this microbiome are quite similar. So you can have different bacteria, but typically, whether it's on metabolic functions or immune regulation, these functions are shared across individuals, and then it can be different bacteria, but these core functions of the microbiome are robustly present across individuals.
Right. So with all of these new findings, then I guess, the idea is that we will see some new targets for diseases. And I know there are a lot of companies out there that are sort of the microbiome companies that are developing new therapies around maybe modulating the microbiome itself and, maybe adding in different strains or removing strains or bacteria. But I understand that Eligo is doing something a little bit different, or actually rather more than a little bit different. Before we get into that, I'd like to ask how you got into this area of work and sort of how Eligo came to be in the first place.
Yeah, so actually, we're not working on the microbiome when we started Eligo. We're working on CRISPR-Cas, and it was like now 10 years ago, a bit more than 10 years ago, when everyone was when CRISPR was just discovered. everyone was trying to understand and see how we could use CRISPR, this new technology, to edit human cells, eukaryotic cells, for gene therapy, gene editing. And basically what we've showed, and we were the first one to show actually that, and trying to see how we could use CRISPR in bacteria. And so what we showed is that actually when you express this nuclease, the CRISPR-Cas nuclease in bacteria, which is basically where it comes from, because it's been discovered as the immune system of bacteria, as part of the immune system of bacteria, prokaryotic elements, cells, then when you create this double-strand break in the genome of a bacteria, as opposed to eukaryotic cells, which have the very efficient machinery to repair double-strand break in their genome, bacteria do not have this, or at least very inefficient ways to repair double-strand breaks. And so therefore, what we discover is that if you express a nuclease in a bacteria and you use this CRISPR nuclease to target a region in the chromosome of the bacteria, wherever you cut, it will lead to the death of bacteria, because it cannot repair double-strand break. And so we thought, oh, this is actually probably the most precise antimicrobial that can exist, because we can kill bacteria based on their genetic signature. And and so we thought, well, that's really interesting. So now the microbiome field was just starting, and we started really by thinking that this technology could be applied to anti-infectious diseases, which it can definitely. But then we thought, actually, beyond infectious disease, because we're discovering that now within the microbiome you have some, and within the specific bacterial species, you can have some strains that are really beneficial for your health, but some that are actually driving disease. And these strains, maybe the only difference they have is the presence of one specific gene, toxin gene, or a gene or peptide that mimics a host autoantigen and drive autoimmune response. And so now we had a tool finally that could enable us not to wipe out a whole species, but even go further than that, actually do surgery in the microbiome by removing just the strains, killing the strains that were driving the disease and not the whole species. And so this is how Eligo came about. And I think beyond CRISPR, what's really important is that, yes, we're using CRISPR, but like for any gene editing company, what's really one of the key limitation or key obstacle to overcome is the delivery. How do you deliver? We know CRISPR can edit genes in human cells, and now we've shown that CRISPR could kill bacteria based on the genetic signature. But how do you get this in a human? How do you manufacture a drug that can indeed have this outcome? And so we spend a lot of time actually, and this is really one of the key know-how and expertise of the company is on building delivery vectors to be able actually to deliver this CRISPR payload to the microbiome, to specific bacteria in the microbiome, to kill them or to edit them or to add new genes. And so, yeah, this is how we started based on this discovery of CRISPR could kill bacteria.
Great. And that was about 7 or 8 years ago the company was founded?
We published we published 2 papers back to back when we were still in academia. I was doing my PhD when I was at MIT, and we were working with a team at Rockefeller University. These 2 papers came out in 2014, and then the company started and has gone now through.
Yeah, so things are going pretty well. So a few months ago, you published an article in Nature showing pretty convincingly, I would say, that you're actually able to go in and select a single strain of bacteria in a living mouse and remove that strain almost entirely. This was using base editing, though. Could you tell us about that story and also the delivery strategy that was developed for that study?
Yeah. So the killing of bacteria with CRISPR, we had shown this in the past already. It's been like 5-6 years that we've showed highly efficient delivery of CRISPR and killing in an animal. But what we've actually thought about is that beyond CRISPR, we know that we can deliver CRISPR and kill bacteria very efficiently. But sometimes you do not want to kill the bacteria because actually, for the whole species carries a gene that is that drives disease, but at the same time, this bacteria is a core member of your microbiome, and you cannot kill it, you cannot get rid of it. And by any means, when you try, it's like there are some bacteria that regardless how much you want to kill them, because they're part of your microbiome, and they're so essential for the function of your microbiome, you cannot kill them. But so some of them can have still some genes that drive disease. And so what can you do to edit this microbiome if you cannot use CRISPR to kill the bacteria? And then, there's been some studies and some discoveries. People have, beyond CRISPR, used, nucleases, but modified version of nucleases to do base editing.
Yeah.
So modify single base pair. And so what we've shown is actually that we can also deliver base editors and use our same delivery vehicle to deliver base editor circuits. And what we've shown is indeed that typically after an oral administration of our modalities, of our vectors, delivery vectors, we can, a single dose can lead to the very efficient and precise addition of a whole bacterial species in the gut of the mice. And this is very stable. So we've shown that this could be done in different bacterial species, but also targeting different genes and editing these genes stably. And so typically, we've done, in the paper, there are a couple of examples where we show that we can, deactivate, we can mutate very specifically a catalytic site of an antibiotic resistant gene, so making it not functional anymore. The antibiotic resistant enzyme or the enzyme, the protein that confers the resistance to the antibiotic, is just not functional anymore because we've edited one base pair, which translates into either a stop codon in the protein or inactive catalytic sites. We've done this on a couple of other toxin genes, typically that are linked to cancer initiation, DNA damage, or even proteins that are involved in the development of neurodegenerative disease. And so now what's super exciting about this technology is that we can now really perform gene editing, correcting the microbiome without touching its composition, and start to address diseases where you do not want to create any changes in this microbiome in terms of composition function. Just edit and have this edit being very stable over time. And beyond the direct applications, and we have some programs, therapeutic programs moving forward, is that it's basically, finally we have a tool. It brings a tool to the community to interrogate the microbiome. It's really hard right now to have good models where you can have a functional microbiomes, and you can until now you didn't have any possibility. There was no possibility to edit this microbiome. You had either to try to put a bacteria that already you had modified externally and then introduce it into the microbiome, which is really hard. But right now we can take a normal microbiome, completely intact microbiome, and try to edit the specific genes that we think are related to a disease. And so by doing this, we can finally really test the if hypothesis where is the gene related to a disease. Let's turn it off without touching the rest of the microbiome, and see if in a specific animal model the pathology is still there. And so that's, I think, really powerful, and actually brings really the tool that was missing to elucidate relationship between bacterial gene and disease very precisely.
Yeah. So it's not just about developing new therapies, but it's also a massive tool for just the research itself, and actually figuring out which diseases make sense to go after, what's most likely to give the biggest impact. So I'd like to stay with diseases for a little while, actually. I think if anybody, searches the scientific literature to figure out what diseases are implicated with changes in the microbiome, I think it's everything. So how does Eligo actually choose which diseases to target? And can you tell us something about the kinds of diseases that you're currently targeting?
Yes, definitely. So I think we're trying to go at Eligo. I mean, we're trying to go after disease where the link between the spectral species and the disease progression trigger is really strong. And this is where there's basically only one bacterial species involved. Because when you start to have multiple bacterial species, then it becomes extremely complex to understand what's happening. And so we're trying to limit as much as possible the biological risk. And so typically our lead program is in moderate to severe acne vulgaris. And what we're going after diseases first because moderate to severe acne vulgaris, it's actually quite really, it has really important unmet need. There's 120 million people worldwide. We're not talking just about a few pimples. We're talking about 40 to 130 lesions on the skin, with only treatments available being either first line antibiotics that don't really work, and the magic bullet being Accutane, isotretinoin, which is an oral retinoid, which has very significant side effects, and people who've been under Accutane will it's interesting when we talk to reporters, journalists, or investors who had acne and Accutane. They're like, "Oh yeah, I understand that it's it would be game changing to have disease." And so we're going after this disease because actually, what's really interesting is that the bacteria that is involved in the pathogenesis of acne vulgaris is a bacteria that is carried by everyone, literally everyone. This is quite important when we're talking about microbiome disease. Often, because the composition of microbiome between individuals varies a lot, sometimes it's really hard to make sure that the species you're targeting is present in everyone. Here, in case of acne vulgaris, we're talking about the most abundant skin bacteria, commensal bacteria, that is really important for immune regulation, immune maturation of your skin, of the immune system of the skin, fighting against pathogens and others on the skin. So everyone has these bacteria, but what we've found, and so in the past years, there's been quite some research literature on the role of and the implication of this specific bacteria in the acne etiology. And so what we have actually seen doing a lot of metagenomic studies is that in acne patients, they have actually a subpopulation of this bacterial species that is overabundant in acne patients compared to healthy patients, and that these subpopulation can be really we can distinguish it from the beneficial C. acnes by the expression of specific pro-inflammatory genes that we can target with CRISPR. And so the whole idea behind this treatment is to have a topical treatment. It's going to be a gel that contains our delivery vectors. The delivery vectors are going to deliver a CRISPR payload in Cutibacterium acnes, the beneficial one and the pro-inflammatory ones. But we program the nuclease to specifically target the genetic signatures that are unique to the pro-inflammatory subpopulation. And so therefore, the idea is to kill just the pro-inflammatory subpopulation while leaving the rest completely intact and enabling actually to correct the dysbiosis, enabling the beneficial C. acnes to take over, and therefore prevent any further or any new lesions from being formed because we just would have wiped out these pro-inflammatory lesions.
That's very interesting. Do you envision a one and done strategy there then, a treatment strategy? I'm just wondering, I don't know much about the topic, but that subpopulation of bacteria that those patients are carrying, that must have come from somewhere. Does it evolve from the parent population, or do they pick it up somewhere else, or what?
Yeah, indeed. So if you look in a household, you have quite, in terms of strains within the household. Indeed, parents, siblings do carry similar strains. But what has been shown also, however, is that so once you have this microbiome, it's stable, but the rate of getting a new strain from the environment from someone else is basically every year you get 1 to 2 new strains. So it's quite stable. You don't get so much. So hopefully we should not do too many. We should not have to do too many treatments, and they will be quite stable. I think right now, if our therapy if you do the therapy for a month to rebalance this microbiome, and you don't have any flares for 3-4 months, that's already a game changer compared to current treatments. Hopefully, it can be much longer. But this is something we'll be able to see, to really understand these dynamics when we go to patients.
Absolutely. That sounds really fascinating. So how far along are you with that program?
Well, it's an exciting time because we're in about 16-18 months, we should be in the clinic in the US in patients, and to try to generate the first clinical data on whether we can edit the microbiome. We can deliver a payload, CRISPR payload, and if you can have an effect on this microbiome, on this dysbiosis, and hopefully try to see sign of improvement in patients.
That's a different route of administration than any of the other CRISPR-based therapies I've heard about.
Exactly.
Is it difficult to develop that formula, or can you learn from the formulation experts for the more conventional topical treatments? How much of the
Yes.
formula is new?
Yeah. So in terms of delivery vector, I think so. We have probably the best experts for biologics formulation, topical formulation around us, and so we're learning a lot from what's done today in terms of topical application. There are few biologics that are used topically, but the thing is, as opposed to the typical CRISPR therapy, because we're not targeting eukaryotic cells, we're not using virus or LNPs, etc. Eukaryotic virus or LNPs. We're using our vectors are derived from bacteriophage, so we're using the structure of the bacteriophage, which is a virus that targets bacteria. Which is protein based, so it's we can lyophilize it. It's very stable at room temperature, and so it's we're working on this formulation, and we're trying to combine both worlds, the topical and the biologic, which is interesting.
Yeah, absolutely.
But, modalities are very small. The hair follicle is actually quite large, and there's been quite a number of demonstration that you can get quite large molecules deep down in the hair follicle.
Okay, great. We're looking forward to more updates on that. So what about any of the other diseases? Can you tell us about any of those?
Yes. Well, I think typically something we're looking at is also the intratumor microbiome.
Okay.
So now it's been shown in different type of cancers and different tumors that they are actually harboring their own microbiome, and that there are some specific bacterial species within this tumor microbiome that actually either act as antagonists to current drugs, or trigger specific pathways, tumorigenesis pathways within these cells. And so something we're really interested in is actually trying to use our modality to deliver payloads into this intratumoral microbiome to either to remove some of these transgenic CRISPRs, or functionalize this microbiome and make it express some ligands or other molecules directly within the tumor to have an effect on the microenvironment. Right. And something else we're really interesting now is that we have this, in gene editing, what's really hard is to get the right vector, an efficient vector to deliver your payload to the right organ. And so right now, we have developed a really good vector for this bacteria in the skin, Cutibacterium acnes, for the skin microbiome. And so beyond killing C. acnes, in the case of acne, we're actually quite excited about the potential of transducing and functionalizing C. acnes directly in the hair follicle, to use it as a small a local production factory for biologics. So basically trying to deliver into this bacteria a circuit that will make this bacteria in their hair follicle express, whether it's nanobodies or chemokines, basically molecules that can have an impact on all the cells and all the immune cells, because the hair follicle is actually a real hub in the skin that controls, regulate the skin immunity. It has stem cells around it. It has a wide variety of immune cells, and so it gives us a really interesting entry point to modulate the skin immunity and enable the expression of biologics that could be quite hard to deliver topically, or that could have actually that do have some significant side effect when they're delivered systemically. And so I think now on the skin and derm, there's quite some potential beyond acne vulgaris, where we kill bacteria, to leverage this bacteria, which in healthy patients is present and really close to immune cells.
Wow, so that has broad implications, I guess, for a wide range of topical conditions. I understand that, one of the biggest issues, I attended a master's course in drug discovery and development some years back, and there were some people from some of the large companies developing dermatological companies, and one of the big challenges is conformity. So people actually applying the creams, no matter how good the formulation's got, it's the stickiness, it's the smell, it's just the hassle of actually having to apply the treatment. So I think anything that can reduce the number of applications would be really beneficial. And I guess that's also something that you envision if you use the vector to deliver other biologics for treatment of other skin disorders. It would probably also be much less frequent application.
Yeah, I think so. For the frequency, indeed, this is going to be something that we'll be able to see in. I mean, the first trial is going to teach us quite a lot about this, because what we know, what we're delivering is DNA payload, and you can engineer this payload to be more or less stable in the cell. And so I think potentially, instead of having to apply a biologic or a cream on the skin, and then after a few hours basically it's wiped out, you don't have anything anymore here, because the bacteria will be transduced with these payloads that they will keep for a while, until it's lost or until they're expelled. This is something also interesting, because we might have a sustained expression of specific biologics. Then for how long, we don't know yet, but this is what the trials and the different models we're putting in place are going to help us answer.
Sure. We have about 4 minutes left. So you started with CRISPR-Cas, and now, the big blockbuster paper that came out a few months ago is base editing. Are you using both of those tools and others in the toolbox, or do you have a preferred gene editing modality, or does it depend on what you want to do?
I think it's all about, we're not technology focused. I think it's more about what is the problem, what is the unmet need we're trying to solve, and how to address it. Whether it is a specific, subpopulation of strain that drive disease. In this case, CRISPR is really fantastic, because it enables us to kill this subpopulation extremely precisely. If we want to do an edit that is stable in the whole bacterial population, we're going to go with base editing. And if we want to have something, a new gene, a new function, integrated in this microbiome, functionalizing the microbiome, we're going to deliver, a payload that enable the expression of the transgene, multiple transgenes of interest. So it's really about what is the problem we're trying to solve, and how to approach it. But what's great is that now we have this toolbox, right? We can do we can do removal of genes, editing of genes, or adding new genes.
Are you working on any programs where you envision an oral administration in humans, so similar to what you demonstrated this summer in mice?
Yes. So we already have our de-risking program in anti-infectious disease, which is IND enabled, which focus on a gut bacteria that is driving kidney failure by due to a toxin expression in the gut. And we have one program in partnership with a top 10 pharma that is undisclosed, that is focused on actually using base editor, delivering base editor in a gut bacterial population. Yeah.
Okay. So just so I don't misunderstand, that has not yet been administered to a human, though, right?
No, it's not been administered.
Okay. How easy do you think it will be I know you can't say until you do it, but can you say anything about how easy it might be to translate that from a mouse to a human?
Yeah, so we've done quite a lot of work, actually. I think it's we've done quite a lot of work with different models, like the SHINE model, that really is a succession of fermenters that have exactly the same condition, pH. They have the human microbiome in different compartments, and that represent change of pH, change of enzymes, change of and length of stay across the different fermenters that represent the whole digestive tract. And we had really good data on that. We managed to create particles that are resistant, delivery vectors that are resistant to acidity, to enzymatic degradation by gut enzyme, etc. So and we've got we have data in mice, but also in rabbits, etc., which really de-risk the whole approach to translating it to human.
Okay, great. I think that pretty much brings us to the end of our time. I think we could have spent a whole half an hour talking about that model. That sounds really interesting. So perhaps you'll come back and tell us more about that another time.
Sure.
Yeah, and we will also meet in April next year in real life for the first time at the CRISPR conference. So I'm looking forward to hearing what you have to share with us at that point. So for anybody who's not aware or would like to learn more, you can just click the QR code at the top of the screen. That will take you right to the conference website. And Javier, please keep us posted on those programs. We love to hear about them, and keep doing everything that you're doing. It's fantastic, and looking forward to see how you completely, transform the microbiome targeting world.
Well, thank you very much, Karen, for giving me this opportunity, and really looking forward to the CRISPR Med conference in Copenhagen next year.
Sure, anytime. Thanks, and also thanks to all of you for joining and for also being patient with our technical delays. Really appreciate that. Hope to see you again.